Hello, I'm Peter Midford, a member of the Bioinformatics Research Group at SRI International. This webinar will introduce you to a collection of transcriptomics data analysis tools available on biopsych.org. Biopsych combines a collection of databases and powerful tools for a variety of bioinformatics tasks. We have 18,000 databases for organisms with sequenced genomes, of which 60 are curated from the literature. The databases integrate genome information with metabolic pathways, gene essentiality data, regulation, and protein features. Our bioinformatics tools support analysis of gene expression data, sequence searching, comparative genomics, and comparative pathway analysis, and the ability to run metabolic models. In this webinar, I will be focusing on biopsych tools for transcriptomics data analysis. I will be discussing importing and working with gene expression data in smart tables, painting gene expression data onto a metabolic map, how to compute highly expressed underexpressed pathways, visual analysis on the Omix dashboard, enrichment analysis, and showing data tracks on a genome browser. Many of these tools are integrated with our Smart Tables facility, so we'll start there. I will create a new Smart Table from the file shown here, which contains gene expression measurements. The data in this file were derived from Gene Expression Omnibus file GSE 71562, associated with a 2017 publication by von Vulten et al. In Genes. The data indicate changing gene expression levels during a transition from anaerobic to aerobic conditions in E. coli. The file contains a list of significantly changed gene expression levels. The data was created by averaging the replicates, then normalizing using the TPM approach. Genes that had zero count in more than 15% of the samples were removed. The gene expression levels were further filtered by removing all genes where the range of expression levels was less than 2 or the p-value was greater than 0.005. We recommend applying this sort of pre-processing prior to loading your data into biopsych. The first column contains gene names, while the remaining columns show expression levels at different time points. To create the smart table, I go to the list of smart tables and choose Smart Table from Uploaded File. Then I specify the file, I specified that the file contains a list of genes in the first column and that the file has column headers. I then click Upload and Create Smart Table. For now, I will focus on the first column, which contains a set of genes, so I will collapse the remaining columns containing expression data. The most basic operation with smart tables is adding columns showing properties of the objects in the leftmost column. So I'll start with a simple example and add a column of left end positions. The column of gene names is selected by default, so all I need to do is choose left end position from the Properties drop-down menu. Likewise, I can add another column of product names for each gene. Finally, smart tables can display more than just text. Here is a column of regulatory diagrams for each gene. Note that each of these will depend on the current organism's database. There may be genes with the same name in other databases, but the results will be quite different. In addition to displaying stored properties of our set of genes, we can create a column by applying computational transformations from a different drop-down menu. Transformations add columns just like properties. For example, we can compute the direct regulators of each gene. This mapping can be accomplished via a transformation. Select the transformation, direct regulators of gene, and it creates a list of the direct regulators. I can go further though by clicking the plus in the last column. It will now create a new smart table containing each regulator that appeared in the final column and all the genes regulated by that regulator. Next, I'll show you biopsych's metabolic map and omics data display. We call an organism's metabolic map diagram in biopsych the cellular overview, a diagram containing all the organism's metabolic pathways and transporters. We can highlight on this diagram all the reactions associated with a set of genes. To do this, I'll go back to the original smart table. Then I will reopen the operations menu, and from the Paint Data menu, I select Paint Data on Cellular Overview. That brings up the cellular overview with reactions that are associated with genes in our SMART table visually highlighted so we can see their relationships within the pathway map. For example, if I zoom into fermentation, we see a lot of reactions associated with genes appear highlighted. This shouldn't be a surprise since this is a study involving aerobic and anaerobic metabolism, and you would expect that a lot of genes associated with fermentation are significantly changed in the original data set, and therefore their reactions appear in this filtered data. If I can zoom in and out with the mouse wheel, if I zoom in far enough, it starts to show the names of some of the pathways. And near the start of mixed acid fermentation is a reaction associated with a gene with changed expression. If I roll over this reaction, a pop-up appears that identifies it. If I click Keep Open, the pop-up window will stay open and I can move it around. And also create additional windows for use in a publication. The T, E, and P buttons within the kept open window let me control how much information is presented. Pop-ups for metabolites work much the same way. I can scroll in further and it will also show the enzyme names and the associated gene name. So we can see for this reaction near the top that the gene that's changed is PFLB. Control panel at the right lets us remove or add back highlighting for multiple sets of genes that we might have highlighted at one time. I can remove all highlighting from the operations menu. Quantitative expression values for multiple time points or experimental conditions can also be painted onto this diagram. For example, I will go back to our SMART table. Then I can choose Paint Data on Cellular Overview Omics Viewer. I select the time point 0 column in the menu, and because these are count values, I will choose the absolute option and click Go. The cellular overview now shows the gene expression levels at time point t equals 0. There's a report window that shows some summary statistics, and I can remove that. On the left is a window we call the Omix control panel. The top part shows a histogram of all the data values in the SMART table and the colors assigned to those values. I can scrunch the second highest breakpoint on the histogram to let us see more variation near the bottom of the range. Now I'll zoom back into fermentation. And if I roll over the same reaction, which now has associated data, the pop-up now has a second button labeled Omix to bring up a pop-up that shows the data graphically. Like the window created with the hold button, I can move these around and keep several of them open. The Omix control panel has a button for removing this data. I can also load data into the cellular overview directly. I will demonstrate this along with the animated display of multi-time point data. Note that I can also load data directly from sources such as the Gene Expression Omnibus. I start by selecting the cellular overview from the metabolism and choose the Upload Data from File command. I then choose the file, which is the same one as I loaded in the SMART table. I then specify that the file contains genes in the leftmost column, that the values are absolute. I also specify that columns 1 through 6 contain data, which are the time point data. Note that this window provides several options for displaying data on the lower left. You can display data on the cellular overview, or as a table of pathway diagrams, which I'll show you later, or as a pathway collage, which is covered in a different webinar, or on the dashboard, which I will cover later as well. Then I click the Submit button. As before, the summary report window appears. If I hadn't loaded this file already, I would want to check it for any processing errors which might be reported in this file. Then I will close the window. As before, I will first scrunch the histogram. Note that the scale is slightly different because we now have 6 time points rather than 1. Then I will zoom into the same fermentation reaction. The Omix pop-up now shows a histogram for all 6 data points, but I can also show the data as an animation. To do that, I'll zoom out a bit. And then go over to the Omix control panel and notice that there is a set of playback buttons that appear because the dataset specified multiple columns. I can start the playback with the play button. And adjust the speed to my liking. Note that there are also buttons to single step. I can also display Omix data on an individual pathway diagram. I'll demonstrate with an amino acid synthesis pathway. First, I'll go to BioPsych's pathway search page. There are a lot of search options, but I'll just enter the name super pathway of L-aspartate, and then I can just choose the rest from the dropdown. And I click Submit Query. This brings up the pathway page. This shows metabolites, enzymes, and genes associated with the pathway. If I choose Customize or Overlay Omix data on pathway diagram, it brings up a dialog. I'll check the box for Superimpose Omix data on pathway. Then, rather than specify the smart table or file again, I'll just use the most recently used Transcriptomics dataset. In addition to overlays, this dialog provides lots of additional customization options. But for now, I'll just click OK. And Omix graphs for each gene with data are displayed next to its reaction. Next, I'll show you several tools that can suggest pathways of interest in an Omix dataset. We can generate a summary table of pathways sorted by their activation levels. One way to do this is to start from the cellular overview, and in the operations menu, load or select some data. I will use the same E. coli time series data for this because it has multiple time points and one of the perturbation statistics only applies to multiple time points. So I will load data from this file. Select that gene names are the kind of data we're looking at. Indicate the data values are absolute. The time series data is in columns 1 through 6. And this time I will, instead of choosing on this diagram, I will choose as table of pathway diagrams. And I will choose to display the top 20 pathways. When I click Submit, the pathway table appears in a new browser tab. This table lists the pathway name, a diagram with gene expression levels at each time point for genes that have data, and a final column that summarizes the genes and enzymes. The third and fourth columns show the pathway perturbation scores. Pathway perturbation scores attempt to measure the overall extent to which a pathway is activated by combining the activation levels of all reactions in a pathway. The fourth column shows a graph of perturbation scores at each time point for the pathway. The third column shows the differential perturbation score, which is a measure of how much the pathway's activation is changed during the course of the experiment. whether from high to low or low to high. It is calculated from the difference between the maximum and minimum activation levels across all time points. The table is sorted by this differential perturbation score. For more detailed explanation, see the BioPsych online help. The Omix dashboard shows a high-level overview of all the systems in the cell. I find the Omix dashboard under the Analysis menu. The command launches a page which lets me select data from one of several sources. I'll take the data from the SMART table with the E. coli anaerobic expression data. I choose the SMART table button and then the desired table from the dropdown. Most of the options default correctly, but I'll specify that the values are absolute and make sure that we are only looking at time point values and not computed columns. I click the Submit button, and in a few moments, the dashboard display loads. Here we see the top-level panels, such as biosynthesis and degradation. And within each panel are finer categories called plots, such as amino acid synthesis or glycolysis. Each plot summarizes the level of one cellular system, with each colored vertical bar representing one condition or time point. For example, in amino acid synthesis, we see the activity for all genes within amino acid biosynthesis pathways at the 6 time points. Display shows expression levels for each gene associated with an amino acid biosynthesis pathway in the small dots. The large dot is the mean activity at each time point, thus depicting the overall activation level of this cellular system. One thing you can do to improve the display is to click on the log scaling button under the display preferences. So you can see the full spread across pathways. If we click on a plot, the dashboard drills down to show a more detailed display. Here we have plots for each individual amino acid biosynthesis pathway using the same conventions as the top level. Each small dot is one gene, and each large dot is the average of all gene expression levels for that time point. If I click on an amino acid such as methionine, I can drill down into it and see expression levels of each gene associated with the pathway. Our next tool, enrichment analysis, is a statistical technique for finding what a set of genes or other objects have in common with one another. That is, are genes from specific metabolic pathways statistically overrepresented within the set of differentially regulated genes in our data set. For example, enrichment analysis could tell you that a set of genes contains more genes involved in, for example, fatty acid biosynthesis than you would expect to find by chance. To perform an enrichment analysis, we make sure that the appropriate column of names, in our case gene names, is selected. Click on the Enrichments menu and select an appropriate set of enrichment analyses. Starting from genes, we could enrich for pathways, transcriptional regulators, or Go terms, or a combination. Each of these are entities that are associated with multiple genes. And combining enrichment over different entities just runs separate analyses for each type of entity, then combines the results and orders them by p-value. I'll use a simple example. Genes enriched for pathways, which I select on the enrichment dropdown, and this pops up a menu with options that control the details of the statistical computation. I'll just take the defaults, although BioPsych provides multiple options for statistical tests and corrections for multiple hypothesis testing. This analysis will create a new SMART table of pathways. Some things to note about this SMART table. The first column contains both individual pathways and pathway classes, which are distinguished from individual pathways by the use of uppercase initial letters. The first row in our table is a class of pathways. The second one is an individual. The third is a class, and the fourth is our mixed acid fermentation pathway that we looked at earlier. The second column is the p-value, meaning the statistical significance of the enrichment. And the column is ordered by p-values, so that the pathways at the top are the ones that are most overrepresented. The last column shows you the genes from the original set that are present in this pathway. I'll go ahead and delete this match column, just so we can see the pathways a little. Shouldn't come as a surprise that the first 24 entries are related to energy production or electron transfer. The first non-energy related pathway is a nucleotide biosynthesis pathway at the bottom of the page. Looking at the p-values, most of the pathways on this page, and certainly the first half of this page, are significantly enriched for the set of genes from our dataset. Biopsych can also display genome-associated data, such as is produced by chip-chip experiments. I'll show you some data from an unpublished dataset from R-Landic that contains chip-chip data for RNA polymerase. I'll select the genome browser. Then I will turn on the tracks display by clicking on the Show Tracks button. This compresses the genome display into a single line and adds commands to add tracks from a local file or a URL. I will load the Landic file. And after it loads, the data for RNA polymerase appears in an external tracks display. Zooming in, the horizontal bars represent the feature data found in the track file and extend from the start position to the end position of each feature. The multiple rows allow overlapping features to be displayed plainly, and the number of rows is of no significance. The score of each feature is displayed both by the color associated with the feature and a graph that appears above the rows of feature bars. I'll scroll over a bit to show some variation in the data. We hope you have enjoyed this tour of the transcriptomics tools provided by Biopsych.